Welcome to Life Science Note, friends. Welcome to another video. So today the topic we will be studying is about endospores. So endospore was discovered by John Tyndall in 17th century, and later it was also discovered by the two scientists which are known as Cook and Koch independently. Endospores are the defensive structures that enable some bacteria to survive in harmful environment. Bacteria produce endospores to survive in harmful environment such as high pH, temperature, acidity, drying out, dryness, chemical disinfectant, as well as when there is treatment with antibiotic. As an endospore, the bacteria is in dormant or inert state. Okay, means in this state it cannot metabolize food, neither it can do reproduction. The endospores are usually produced by gram-positive bacteria. Mainly, the common bacteria which produce endospores are Bacilli and Clostridium species. The other bacteria which produce endospore are Sporolactobacillus, Sporosarcina, and Desulfotomaculum. If we see an endospore under a microscope, it is like a bacterial cell, and here is your endospore after the staining technique is done. So the location of the endospore also differs among bacterial species, and it is useful for the identification. There are three types of location of endospore inside the bacterial cell, of course. The first is terminal, which is the endospore at the end pole of the cell. The second is subterminal, which is near the end pole, okay, close and near to the end pole. The third is central placed endospore. The central placed endospore is in between the cell. The example of the terminal endospore consisting of bearing bacteria is Clostridium tetani, which causes the disease tetanus. The central placed endospore consisting bacteria is Bacillus cereus. So the positioning of the endospore differ among different bacterial species and is useful for the identification of the bacterial species which is producing the endospore. Now let us see what is the structure of endospore. Endospore consists of an exosporium, a spore coat, and an inner coat. Apart from that, it also contains a cortex, which is DPA and calcium rich, which give its turbidity. It also consists of core wall, inner membrane, some DNA and DNA binding protein, as well as some ribosomes and some enzymes which are necessary for the germination of endospore to the bacteria. So now we'll see step by step the layers of endospore and what does these layers consist of. So the first one is spore coat. Spore coat is an electron dense layer formed outside the outer membrane. It is made up of keratin-like protein and other spore-specific protein. These spore-specific protein give hardness and rigidity to the endospore. The spore coat is also rich in cysteine and hydrophobic amino acids, which are later used while the germination process. These cysteine keratin proteins give resistance to the chemical treatment. So the second layer the endospore consists is the inner membrane. It is also known as germ cell membrane. So it basically just surrounds the cytoplasm, means in which the DNA and the other binding protein and the ribosomes are. So it just surrounds the core of these endospore. So the third layer is the cortex, which is this dense layer. The cortex develops between the outer and the inner membrane of the endospore. The cortex consists of layer of peptidoglycan, which consists of two layers, outer layer and the inner layer. So the outer layer is loosely cross-linked while the inner layer is tightly cross-linked. So as I have said earlier, the cortex contains the peptidoglycan layer, which consists of the two layers, outer layer and the inner layer. The outer layer is loosely cross-linked while the inner layer is tightly cross-linked. The inner layer contains three repeating subunits. The first is alanine, second is tetrapeptide, and the third is a muramic lactam. The aniline subunit is 55%, while the tetrapeptide subunit is 15%, and the muramic acid subunit is 30% of the total inner layer. The alanine is attached to the L-alanyl residue, while the tetrapeptide bears a sequence of L-ala, D-glutamic, meso, DAP, D-alanine. While the muramic lactam does not have any amino acid attached to it. The cortex also contains DPA, which is adipocholic acid, also contains some calcium ions. The structure of DPA looks like it's a benzene ring attached with the carboxylic group and nitrogen group. The fourth one is the core. It is also known as protoplast. So the protoplast consists of a DNA, the SASPS proteins, which are small acid soluble spore protein. What these proteins act as, they protect the DNA from the UV radiation. They protect the DNA from the UV radiation as well as the heat. The next is, there are some protein synthesis machinery available in the protoplast itself, which is a ribosome, tRNA, and some enzyme. These enzymes help the endospore to germinate. I hope you all liked my video. Please don't forget to like, share and subscribe to my channel, and I'll see you around in my next video. Thank you.